So good morning students. In the revision for strategies for enhancement in food production, we had discussed 1 mark questions and 2 mark questions. So today we will see about the 3 mark questions. What are the questions which we will ask for the 3 marks? Okay. Yesterday I had given some of the questions. Today I will explain the answers of that particular questions. Okay. Let's see here. In this 3 mark question, what questions will be asked with that is explain what is meant by biofortification. Explain what is meant by biofortification. Okay, here first we have to mention what is the meaning of biofortification. So let's see here. Biofortification means breeding the crops with higher levels of vitamins, vitamin A especially, vitamin A, iodine, zinc, okay, higher level of vitamin A, iodine and zinc, okay, which increase the risk for disease. See here, first of all, vitamins and minerals, we can directly write it as the vitamins and minerals. Okay, then are higher proteins and healthier fats. The next here it is the most practical means to improve the public health. Here, what in which topic it will come? The term biofortification means it comes under the plant breeding for. It is one of the aspect of plant breeding for improved food quality. Here, whatever the plant breeding technique is there, that is practicing for what means to improve the food quality. In what sense we are telling the food quality means see the food which will have the nutrients such as the carbohydrate, proteins, fat, minerals, vitamins, everything. Okay, see due to the increasing population, due to the production of hybrids, due to some of the plant culture and all, here there is a diet in a human beings, whatever they diet, they lack the essential micronutrients as well as the macronutrients. Okay, what is that micronutrients means? Which the nutrients which is present in a very lesser amount, as well as the macronutrient means which is present in a very higher amount. So if the food does lacking that all nutrients means that human will suffer from the absence of nutrition or reduced amount of nutrition. So what happens means if such type of essential micronutrients are lacking in the diet of a human beings means which will lead to especially that is iron, vitamin A, iodine, zinc. If these nutrients are lacking here means that person may suffer from many diseases. Okay, and that condition we can say it as the malnutrition. You can see in the slum areas, whatever the areas, the small children or the human beings which are present, living in the slum areas, begging for the food, they lack the nutrition. They will not get the sufficient food. See, everyone cannot buy daily apples or daily fruits. Okay, so some can buy, some cannot buy. So how their nutrients are going to be fulfilled? They had given one thing that to come over that problem, they had discovered this plant breeding technique for what? For improved food quality. So whatever for the improvement of the food quality, in what sense means in the sense of nutrients. So what is the meaning? It's what they had given what term means, that is the biofortification. Explain what is meant by biofortification. Biofortification means the breeding. Breeding by what? That is plant breeding. Through the plant breeding technique, they are going to breed the crops with the higher levels of what? Especially that is the micronutrients. Okay, which is required in a minute quantity such as vitamins, minerals, higher proteins and healthier fats. So that is known as the biofortification. If they ask for 3 marks, whatever the meaning is there, that will give me 1 mark. Then another 2 marks they are telling what are the objectives for this improving the plant breeding, for improving the food quality means what you have to write means see here. Objectives: breeding for improved nutritional quality. Breeding for improved nutritional quality of food. Nutritional quality of food with the objectives such as the first objective is protein content and quality. Objectives: improving. What should improve in that diet means here there are some objectives. The first one is improving the protein content and quality. Protein content and quality. This is the first objective. The second one, that is oil content and quality. Oil content and quality. This is the second objective. The third objective is vitamin content. And the fourth objective is micronutrient and mineral content. Micronutrient and mineral content. So these objectives will carry 2 marks. Okay. In what sense they are improving? By improving certain objectives. For what? For improving the nutritional quality of food. First one, they have to improve the protein content and quality, oil content, vitamin content and micronutrient and mineral content. So this answer will carry how much? That is 3 marks. 1 mark for meaning and 2 marks for objectives. Okay. The next we will go for the next question. Here, sometime they will ask example, mention the example of biofortification. Here they had given, I will write here, example for improved food quality. If they ask like this, means example for that, Atlas 66, which is a wheat variety. Example, Atlas 66, which is a wheat variety. In what sense they had improved the quality of this wheat variety and they had given the name Atlas 66. Sometimes they will ask for 1 mark, like the questions. For 1 mark. Okay, what it is Atlas 66 wheat variety in which they had improved the protein quality, having improved or having a higher protein quality. It has an high protein quality. Okay, the next one more example is there, that is iron fortified rice. That improves the quality of iron in what? In the rice, and they had given the name as iron fortified rice. These are the two examples of what, and many other examples are there that you can refer your book. The next we will go for the next question. Here the next question is, what is SCP? Second question, what is SCP? Question mark. Okay, the next explain microbe used for, explain microbes used for the production of SCP and mention its importance. Mention its importance. Okay, this will be the question. First, we will see what is SCP. That is single cell protein. Single cell protein. Okay, it is, what is the full form of SCP? Means that is single cell protein, refers to the production of refers to the production of edible proteins. Production of edible proteins. Okay, that is the production of what? Edible protein. Edible means which can be eatable on a large scale by use of by means of microorganisms. Okay, by using or by utilizing some of the microbes, they are going to produce the edible proteins. Okay, and what are the microbes which are used over here? So, here some of the common, here what they have told, explain microbes. What are the microbes which are used in the production of SCP? Here, such as some of the common microbes which is used in the production of SCP. First one, that is bacteria. In that bacteria, Methylophilus, Methylotropous. Which bacteria? Methylophilus, Methylotropous. This is one bacteria. This is one microorganism. Second one, cyanobacteria. Cyanobacteria. What is the example of cyanobacteria? Spirulina. Okay, the next that is yeast. Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, the next algae, Chlorella. This is about the microbes. This is the meaning. 1 mark, 1 mark. Then any two also you can mention. Okay, no need to mention all the 4. Any two you can mention if they ask for 3 marks. Then the last one, mention its importance they had given. Okay, what are the importance of this SCP? How it is useful or how it is improving the food or what are the advantages of SCP? Let's see here. Importance of SCP. Importance of SCP. Okay, what are the importance over here? So, let's see. High quality protein and less fat content. Whatever the single cell protein, by using the single cell organism, they are going to produce the edible proteins. The proteins will have a high quality protein. First point. High quality protein and it will have a less fat. This is one important. Then reduce pressure on. It reduces the pressure on. Second one, it reduces the pressure on agricultural production system. Production system. This is the second important. The third important is that minimizes the environmental pollution. Environmental pollution. The next can be produced throughout the year. It can be produced throughout the year. So these are the importance of what? Importance of SCP. If you mention any two, you will get a 1 mark. 1 mark for meaning, 1 mark for microbes, and 1 mark for importance of SCP. Okay. Here, high quality protein and less fat. It reduces the pressure on agriculture production system, minimizes the environmental pollution, and it can be produced throughout the year. Then we will go for the next question. The next question, explain. See, this question is for 5 marks. Explain Moid technique. Explain Moid technique for technique of hybridization. Sometimes they will ask explain the steps involved in the Moid technique. Okay, in that first we have to expand the Moid. See, first of all, Moid means that is multiple ovulation and embryo transfer. Okay, this is the expansion of what? Moid. Okay, this is also called as the superovulation. This, if you want, you can write, no need. This is also called as the superovulation. But also I will here mention it. It is also called as superovulation. Okay, superovulation. Here, this technique of hybridization is mostly used for cows. In this technique, the cow is forced to ovulate more number of eggs so that eggs can be fertilized for the production of more number of cattle. Okay, so directly we will go for the steps which are involved in the moid. Here it involves the 4 steps. Involves the 4 important steps. In that, first step is in induction of superovulation in donor cow. This is first step. Second step, artificial insemination and recovery of embryo. This is the second step. The third step is a transfer of embryo. And the fourth step, that is a pregnancy diagnosis. These are the 4 important steps. See, if sometimes they will ask only write the steps involved in the moid technique. Sometimes they will ask explain the steps involved in the moid technique. Okay. If they ask to explain the steps in the moid technique means at that time you have to explain each step. In the first step, induction of superovulation in donor cow they have given, you have to mention about the drugs or the hormones which they are using. Here that is FSH and prostaglandin. Which hormone they have used? FSH and prostaglandin hormone they had injected into the donor cow. Okay, and that FSH is administered for 4 to 5 days, and whatever the prostaglandin hormone is there, that is also injected for 3 to 4 days. If you mention this much, you can see, due to which there is due to the injecting of these hormones, there will be superovulation is going to pass. Superovulation means that is around 6 to 8 eggs are going to produce. And there is a production of 6 to 8 instead of 1 or 2 eggs. If you inject with these hormones FSH and prostaglandin, that will be for a very a specific time duration. If you inject that hormones, that particular donor cow will produce, instead of 1 to 2 eggs, they will produce the 6 to 8 eggs in their or in per estrous cycle, and that process is known as the superovulation. Then second one, artificial insemination and recovery of embryo, which is the second step. Here in this only you have to mention the donor cow is artificially inseminated with the semen of the male bull, okay, which is collected from the superior quality bull. If you mention this much, okay, over. The next, transfer of embryo. In this transfer of embryo, the recipient cow should have a same reproduction cycle, that is the estrous cycle, with the donor cow. Here, in this transfer of embryo, it should be, care must be taken to see that whatever the donor cow and the recipient cow will be there, they both should have a same reproductive cycle. This is the first thing you have to mention. The donor cow and the recipient cow should have the same reproductive stage or same reproductive cycle, and whatever the technique here they had given, that is transfer of embryo. First, the embryo is transferred to the uterine through a particular instrument that is known as the catheter. That much enough to write here. Then the last one is pregnancy diagnosis. The pregnancy in the surrogate cow can be confirmed by palpation. By which method the pregnancy can be confirmed? That method is known as the palpation. Palpation means by inserting the hands, okay, by inserting the hands in through the rectal walls between 1 to 3rd month of between 1 to 3rd month after embryo transfer. During 1st to 3rd month after embryo transfer, by inserting the hands into the rectal wall of the surrogate cow, can diagnose the pregnancy in the cow. So this is about the moit. Explanation about the steps of what? Moit. The next we will go for the last question. Explain the steps involved in the plant tissue culture. See, in the plant tissue culture, only they had explained the steps. Okay, if they ask only steps, you can mention the steps, that is collection of variability, evaluation and selection of parents, cross hybridization, testing, release, and commercialization of new cultures. So this much if you mention, that is enough. Okay, so thank you. So tomorrow onwards I will continue with the microbes in human welfare. Okay.